Hello, everyone. Thank you for joining today's Codex workshop. I'm Amy Sterling from Princeton University, joined by Arie Matsliah, also from Princeton, from Sung and Murthy Labs. And Arie's really been the mastermind behind Codex, that it would not be what it is today without feedback from the community and the users who are in Codex and making use of it for their research. So we're really grateful for your use of the platform and also for your feedback and suggestions. So I just really quickly, before we go into the Codex workshop, wanted to give a brief update on the state of proofreading in the bank, the brain and nerve core data set from Wei-Chung Lee's lab. I know a lot of you guys have already been using it in Codex, but proofreading is looking pretty good so far. We've got around 90% of neurons in the central brain and VNC have been proofread as well as the neck. We've been focused on the right optic lobe, so you can see there's still some work to be done in the left. And the synapse, true synapse rates are looking pretty good for the bank as well. And here's an update. This is from Alex Bates at Wei's lab at Harvard Medical School. Just an update on the interdataset matching. So the VNC, about three-quarters of the neurons have been matched, verified, and the other, another roughly a quarter have a high in-blast match. The central brain, still more to be done, but there's a pretty good in-blast matching for a good chunk of the cells in the central brain. And this is just a snapshot of kind of how the assignments are put together and where the in-blast matching kind of is falling on the spectrum here. So looking pretty good. And then lastly, just kind of this diagram showing where the proofreading where the annotation has been focused. So you can see not much going on in the left optic lobe, but kind of focused on the right, BNC and net connective, and on the midbrain as well. So that's just a very brief update on the status of BANC, which, as you know, is in Codex, and there should be a paper coming out, preprint coming out soon. But so with that, I want to pass it off to Arie so that because we've only got 30 minutes here to talk more about Codex and answer the questions that you guys submitted via the RSVP. And if you guys have any questions, feel free to type them in chat or just, I don't know how to do the raise your hand feature, but you could try that and we'll try to answer it.
Thanks, Amy. Can you hear me okay?
Yes.
Okay, so I reviewed the responses from the survey. Thanks for providing the feedback and requests for clarifications. And I made a few slides just to go over the most common questions or topics. And it will be mostly me demoing on the codex itself, how to do certain things. Throughout, I will also highlight some of the new data sources and features that came to Codex. So as Amy mentioned, in addition to FlyWire, which is the flagship data set behind Codex, we also have BANC in the Codex. It requires access tokens. So if you want to access BANC and you don't have the token yet, you will need to fill out a form. There is a link and you will get access to this data set. It's not fully proofread yet, but it is mostly there, and it's already pretty interesting. Then, in addition, we also added the MANC male nerve cord data set into Codex. And soon we'll have the optic lobe also from Janelia, the male optic lobe data set. And all these four data sets, the idea to have them tightly integrated so you can quickly jump from cell types in one data set to another data set. For now, you would switch over from one data set to another here. For instance, I can come here from FAFB to BANC and to MANC, and all of these are kind of separate apps for the separate data sets. We are also started working on two mouse data sets. One is retina and one hippocampus, but these are coming later once these data sets are published. These are both being worked on in Song Lab right now. Um, So the most common questions were a lot around the query language, and I wanted to quickly go over. This is not new, but I want to quickly go over some of the common ways to form queries. First, to clarify, the query language is pretty much identical in all of these different applets that you have. The search bar in search, or whether you're looking for cells or 3D visualizations or connectivity and pathways, all of those search boxes, they basically parse the same, use the same query syntax. By default, the query is matched against all the different annotations that we have. So if you type in any free-form text, it will be matched against structured annotations like cell types, superclasses, and subclasses, and so on, but also against the community labels, which are more like free-form text and sometimes not out of date and so on. If you want to be more specific, then you will need to start building these structured queries. And then here, the structured queries are they somewhat resemble the way you would kind of form a query in a programming language. There's a notion of attributes. These are the things that are attached to neurons or connections between neurons. For example, superclass type, cell type, and so on. And there's a notion of operators. These are typically in codex to separate attributes from operators. We mark the operators in these curly braces. Okay. And the examples for operators could be something like has. For instance, you can check for, I want the cell type that has the nerve annotation. So I would do something like has nerve. And this will give me all the cells. So we have about 10,000 cells with nerve annotation. Similarly, for other unary operators, you can, this is the opposite missing. You can ask upstream, so upstream of a certain cell, or a similar connectivity to a certain cell. And then there are also binary operators where you would have provide two arguments to them. And the most trivial ones would be equals. So for instance, you can ask cell type equals, let's actually do that. And there are shorthands for these operators. For instance, I ask if cell type is MI1. And so I get all the different neurons where cell type equals MI1, but it's not gonna match neurons where cell type is not MI1, but there's MI1 in the community label. Right? So here you get 1,584, but without this, I probably get more because it will match everywhere. Right? So you see all of those cases. And then let's do an exercise here. So you can also ask, so here in this query, I'm saying match everything with MI1, but also where cell type is not MI1. So I want to see where are all those cases where MI1 returns a match, but it's not because of cell type. So if I run this query, then you see it's just prefix matches for MI14. By default, it will just almost do like a find in text. This is the kind of the behavior for the freeform queries. Then on top, there is a third type of operator, and these are the logical operators. And this is where you have two queries or more than one query, and you want to combine them. So you can say match certain query and another query and another query, or and not, and so on. So again, like in a programming language. And in those cases where you're assembling elaborate structured query, you can group the different parts using these double curly braces. So for instance, I can do something that matches query 1 and it doesn't match query 2. So this will be the syntax here. My first query would go here, my second query would go here, and it would match everything that has this but not this. There are links provided here, and we will share this deck. So in the FAQ page, there's a big section about the query language, and it has description of all the different attributes that are available to search, as well as all the different operators, binary and and unary, as well as the logical operators. And then there is also a section that's linked from here for sample queries. So these are kind of starting from very easy, going to a little bit more elaborate sample queries that you can try. And when you try, you can then adjust the query and play with it and just to learn on the fly how the query system works. The next set of questions were about 3D visualizations and being able to customize the 3D views. So here again, maybe I'll just do the demo. So the easiest kind of case, is there a question online?
It was just about bank access. I'm sending it to Marissa now.
Oh, okay, all right. So yeah, so Amy, if there are any questions, please stop me. Um, so the easiest is just a single cell. Yeah. So here I provide a cell ID, and this is the cell, and then it has input and output synapses marked with these markers. So I can. This is NeurogLancer, obviously, and then I can visualize these markers for input and output cells. Then there is also ability to, if I'm interested in all the upstream and downstream partners of this cell, with a single click, I can load them. So this cell has 95 partners. Let's see. All right, so you see this is the cell of interest, the one cell that we queried, and then I can switch off. So this will be all the upstream cells, and this will be all the downstream cells. And these will be cells that are both upstream and downstream of the cell that we queried. Now, in addition, for groups of cells, for instance, let's take these cells. There was a questions about assigning, color coding different cell types, for instance. And this is pretty easy. You just select any of the attributes that are available in this list, and it will assign color based on that attribute. So in this case, I'm coloring them based on the type, cell type. You can also change your color scheme if you are looking for, making screenshots for your figures. And you can also load electron microscope images if this is something you're interested in. There was also a few questions about how to share links to 3D viewers. So this share function here doesn't work. This is the NeurogLancer share because we do not require signing in and using the state server. However, if you just copy these links, and these links are typically very short because all the kind of resolving from types to cell IDs happens on the codex side. So these cells are persistent. So if you just share this cell with anyone that has a codex account, they will just see exactly the same view, same configuration, not the same angle, but the same configuration. All right, so let's see what else I had there. Yeah, I think I mentioned everything here. Similarly, there were some questions about network visualizations. I want to do a live. So this is the sample network that we show in the network applet. There are a few things you can customize here. So, but before I go to customizations, I want to mention if you are looking to make figures for your papers from this network views, I would say most likely you will want to download the data as a JSON and load it in something like Citascape. Citascape is or any other app that you like to edit networks. And this JSON file will contain all the information that is available about these cells and connections that are shown here. But still, for quick exploration, you may want to play with some of the settings here. So these labels on the nodes can be adjusted. Here I'm showing the cell types for every node, but instead I could show cell name or cell ID. So if I just click that, the labels change. Then I can also group the cells by cell type. Let's see. And then instead of having one node per cell, now I have one node per cell type. And you see this node, for instance, contains 2 cells. And these are cell types that only contain 1 cell within this query. Then there are additional, for instance, if I go back to individual cells, I can show Brayton regions, and also I can include partners. Let's do just this for simplicity. I can include partners of all the cells that were queried. So now these are the 5 cells that were queried, but I'm also showing all their upstream and downstream partners as well. There are additional settings here to explore, like thresholds on the connectivity strength, as well as how many connections to show. It will basically use this parameter to show the top connections in terms of synapse counts. And then there is also a way to adjust the layout. There are a few options for the layouts implemented, so you can see the one that works best for you, and then you can move them around. And but again, as I noticed, in most cases, if you want to make a nice high-resolution figure for your paper, most likely you will start here for exploration, but then download the data and load it into some tool that is dedicated for making figures. All right, so next thing I had was references and external links. So we made quite an effort to interconnect the data on Codex with the different resources that are available and built by the rest of the community. Some things that you are probably already noticed. For instance, we had these links for some time to Neuron Bridge to find driver lines, and the links to Brain Circuits IO for matching genetic lines. So these links take you directly to the right place for this cell. Then recently also, this is still somewhat work in progress, but information that was provided by Claire, I don't know if Claire is online. We have now links to virtual FlyBrain and FlyBase databases. So again, I have some pre-made links here. In this case, if I click this little links section here, then I can see the same neuron in virtual FlyBrain. Let's try this. So this will take a moment to load. Now, this is the exact same cell in virtual FlyBrain, and you can get a bunch of additional information from here. You can also see the cell type in FlyBase and the information that's available on this cell type. And there is also links extracted from these community labels. So for instance, in this case, someone from the community noted this paper, so this paper is also linked here. There is also automated cell type informations generated with LLMs. These are kind of, again, work in progress. Not sure yet if they are valuable, but you're welcome to try. This will use the database behind Codex as well as the information online to give you additional information about these cell types. Sometimes these references are somewhat bogus, so use it with care. Then also very exciting coming soon, there's effort led by EJ, and she's basically curating a list of known functions for cell types. And we're planning to incorporate it in Codex. This should be available maybe next week. So you'll have known functions for cell types. Okay, there was a set of questions also about finding similar neurons. In Codex, there are three primary ways to find similar neurons. If you're looking for a certain, if you find a certain cell and you want to find all the similar cells, you can use morphology. You can use connectivity or connectivity on level of types. And so I want to demonstrate this quickly. So for instance, here's one use case. You would want to annotate some cell that has no type yet in Codex. So we have about 1,600 cells in Codex without the type. And so let's say in this cell has 10 cells with similar shape using NBLAST. So we can see all the different cells with a similar shape, and we see that two of the top matches are of the same type, CB1508. Let's see down. Yeah, and there's another one. So there's a good indication that this cell probably should be of the same type as well. But then you will probably want to also cross-check it with connectivity similarity metrics. And here you have two ways basically to compare connectivity, one based on the individual partners and the one based on the types of the partners. And then in each one of them, you can look for both up and downstream similarity, only upstream or only downstream. So in this, let's try this, for instance. And this doesn't give us the interesting result, but let's see. Let's see, maybe. Yeah, this one looks more related. So again, this CB1508 comes up here as well. So here as well. So in this type of kind of analysis, I would conclude probably the most likely type for this neuron type for this new neuron is CB1508. Now, while for many neurons, the similar shape neurons are not available, the connectivity-based similarity is always defined and available. So for instance, let's say in this case, there's no matches in NBLAST, but I can still find something similar using connectivity only. So I would say so this is the most similar partner to this one based on connectivity. Okay, so I want to conclude on this here and move to the next section. Yeah, so there were a bunch of questions on programmatic access. The short answer is that there is no plan yet to open up API access to Codex. There is two alternatives for now. One is you can download the data. There's a portal with all the different resources that are used behind Codex. Each one of them is defined. Most of them are in simple CSV format. You can just download and use with any tool. Then there is also a set of applications available on our FlyWire homepage for working with connectomic data. And here you have a bunch of applications like Coconut, Fly, and others to fetch data, Cave, obviously, to fetch data from databases, from live databases, and use in your own programs. I do want to highlight, though, this came out recently, comment by Meet. The data here now reflects exactly what is served in Codex, while many other apps, they look at other snapshots of data. So for instance, Cave is proofreading database primarily. So you will have the information that was fed into Cave, but in Codex, we also pull in information from other sources, like new papers that come out online, and for instance, things that I mentioned earlier, links shared by Claire. So from there, we can infer other cell types and annotations. So all this is maintained by us, and this is probably most up to date. So if you want to get the most up to date data that reflects Codex, you would come here and download the data.
Hey, Arie, there's one question from Meet Zandawala.
Yep.
Can we find neurons based on soma location? Specifically, if you have a rough idea of the brain region where the soma would be found, is it possible to build a search box and reveal neurons in that area?
Yes, we could do that. There is soma position is served, and it's also indexed. So we could do soma location based search. Should be straightforward. Can you take a note of this, and we can think about how to
Yeah.
do it?
Yeah.
Yeah, we need to. I mean, we have plenty of suggestions for new features and additional data. We need to somehow prioritize them and work on them in our pace. But yeah, all the suggestions are very welcome. Okay, so I had so we have maybe 3 minutes for Q&A. Happy to take questions live or any other discussion topics.
Hey, Ari.
Hi, Mit.
Uh, Thanks for answering that question. I had another one. And this is probably not just relevant to Codex, but also talking about SOMA locations and tables indexed elsewhere. So we've noticed that in cases where the nuclei has not been segmented properly, the algorithm assigns SOMA location to be the end of a neurite branch or something, which is not exactly true. So for a lot of, not lot, couple of thousand, I think it was about 7,000 neurons, we don't really have a SOMA location. Are there any efforts going on to manually identify these so we have a better idea?
I'm not aware of such efforts. Worth asking who would be. Maybe, I mean, we need to do some asking around. I'm not aware of anyone looking for missing somas.
Yeah.
Okay.
And also, do you know for those cases if somas do exist in the data set?
I can send you separately the
Yeah,
details of our analysis, and but it's about 7,000 neurons that we found that don't have nucleus predicted.
I see. I see. Yeah, I'm not aware of such an effort.
Okay.
Hi, Ari. I have a question about
Hi, Ocean.
hi. I have a question about Cave. So I wonder how up to date it is. So if I download the neuron IDs from Codex and then I load them in Cave, and then I look up these neurons connection, uh, can I trust those connection?
Yes, connections you can trust today. But again, this is also being updated.
Okay.
We have just predicted new set of synapses and looks like they are of improved quality, improved accuracy. And once we conclude that they are definitely better than what we have today, we will swap the connections in Codex, or for some period, we'll probably serve both old and new.
Yeah,
But as of today, the connections should be exactly in sync in Cave and in Codex.
I see. Okay, thank you. And what's the version that there's a version in Cave that I can print out then?
Yeah, 783. Yeah, these are also in Codex. When you on the main page, it shows the version of the dataset.
Okay, got it.
So
Thank you.
in Cave, you can just specify this. It's called materialization version,
Yeah.
and then it will give you the
Okay,
snapshot.
got it. Thanks.
Hey, I have a question about the versions.
Hi. Sorry.
If I'm going from 630 to 783, as long as the root ID is the same, are those two neurons the same?
Yes.
Okay. Are I also know that the synapses were updated. So would the new neuron have different synapses than the 631?
Um, so yeah, so the synapse updates are kind of orthogonal to the segmentation, to basically to the IDs of neurons. And I'm not sure if we are going to map the new synapses to 630. Most likely, if you can make the effort to upgrade to 783, which is the official publication version of the
Mm.
segmentation, it would be worth the effort. And we have tools to map all the IDs. So one of the kind of obstacles people have with moving from older versions is this mismatch in IDs for the same neurons. There is a tool in Codex where you can plug in 630 IDs that are not in 783, and it will give you the right, in most
Mhm.
cases, the right 783 IDs.
Yeah, okay. Yeah, I've seen that one before. Thanks.
I think we're out of time. Uh, we can send this slideshow, and also happy to get more feedback and suggestions.
Yeah. Thanks everybody for joining. We'll send a follow-up email, like Ari said, to everybody who RSVP'd, including a link to the recording and the slides. And if you have any questions, feel free to ping us, ping Ari. And thanks for being here. Thanks for your time.
Yes, please ping on. There is a link to Slack channel on Codex right top corner. So join the Slack channel, and then you can have direct communication.
All right.
can have direct communication.
Right.